In Germany, biodiversity is, very low. There's not much there. It's Northern Europe. So I wanted to study abroad somewhere, and I went to South Africa. And I studied a year in South Africa at a little museum. University, but there was a little museum. And the director of that museum, Jason Lund, is the expert of African assassin flies. So I worked with him. We published 2 papers during that year that I was abroad. And when I was there, I learned about an internship program right here. And luckily, they took international students, so I actually worked with my predecessor, who was the curator in charge of flies before, in the year 2000, and I was an intern. And at that internship, I got to know my later PhD advisor at the museum in New York. And so it all sort of came by leaving Germany. I traveled the world basically to end up here. Very early on in my sort of scientific career, I should say, when I did my basic biology degree back in Germany, I wanted to work in a museum. And that could have been a university that has a big collection, insect collection in my case, or a museum such as this. And so when I started my PhD at Cornell University, which is has one of the biggest insect collections for a land-grant university, not a private university, I did part of my PhD at the American Museum of Natural History in New York City, which is the second largest museum in the US. And then for my postdoc, I went to the Field Museum of Natural History in Chicago. And now I'm here in the biggest museum. So I've gone everywhere I went with just museum collections. So this is my dream job, to be, curator of flies at the Smithsonian. So that 2009 article was part of my PhD dissertation, in which I tried to decipher the evolutionary history of these flies. They're called assassin flies or rubber flies because they're predatory and they kill other insects. They don't do anything to us, but they like to eat other insects, even some spiders. And there was nothing like this study before in this group of flies, which is about 7,500 species that are known from around the world. With the DNA study, it really depended on my fieldwork and other people sending specimens. I did not use museum collections, a pin fly, to extract DNA from those, because my study was really relying on nuclear protein encoding genes, and the chance at the time, this is 10 years ago, that I actually gathered the data, of getting high quality DNA sequences from pinned dried specimens that might be 20 years old was just not good enough. And so now there's some ways or some new techniques that you can actually go back, use these old specimens and get maybe good molecular data as well, but at the time that wasn't available. In a way, in molecular data, you learn the lab techniques which you can apply to flies, to beetles, to birds. There's not much difference between whatever organisms you use. And so in a way, it's a little easier to use those. However, you don't have as many specimens available. We have in our collection here about 75,000 specimens of these assassin flies, and I have assembled the largest collection of these flies preserved in ethanol for DNA extraction, and maybe I have 500 specimens available. So it's a very different basis of specimens that you can work with. So morphology is fun because you can get every single one you want because it's in some museum around the world. And for DNA studies, you really have to go either catch them yourself or be lucky that somebody sends it to you. For me, the pinned collection like we have here haven't really played a big role in molecular research, and that's mostly because I wanted to cover the evolutionary breadth of these flies, and they're about 120 million years old, so going back to the dried specimens might not give me enough data. But if you do population genetic studies, they would have been very appropriate. However, now we have at this museum the biorepository, which is sort of a liquid nitrogen tank genomic collection, and I'm actively adding specimens to that collection. Actually, we're sequencing the first genome of an assassin fly based on collections that I put in there. And so that's like a different kind of collection, but it's the same. I mean, I put all the flies that I particularly preserve for this purpose in there. And so somebody in 200 years might be able to take one out of the tank and sequence a genome of a fly that that person might be interested in. So historically, the collections weren't so important for me for molecular research, but going forward, it will be a great resource.